Hi, everyone. So, as was mentioned, I'm Kennedy Schaal. I'm CEO of Rejuve.Bio. We're part of the SingularityNET ecosystem. Let's see, is this my slide thing? Okay. So, why curing was not aging was not easy until now. It's a little bit of a tongue-in-cheek title of this slide. Obviously, prevention research has been severely lacking. Of course, in the United States, the way that our healthcare system is structured is that chronic disease makes money and cures do not make money. So we are at the mercy of treating people once they are already sick. Anti-aging research is pretty slow. Humans obviously live too long to study in a laboratory sense, and most common animals that we use to study aging also can live years, such as mice and rats. It's rare to see cross-data set analysis combining and integrating human genomic data along with animal genomic data. Personalized medicine is a huge untapped resource. As was mentioned before, aging is a multifaceted problem. It's going to require a multifaceted approach to treatment. Some of the novel points that Rejuve.Bio is using to try to crack this code of aging is our novel AI technology. We have a sophisticated knowledge graph, which is a recursive metagraph and allows simultaneous application of multiple AI techniques. And what's really cool is if one AI technique kind of gets a little bit stuck in data analysis, there are other AI techniques that can take over. We have our Bioatom space, which is our AI platform, and has multiple interconnected neurosymbolic processes that all work together, and it's continually updated with new data. Being part of the SingularityNET ecosystem, it's a decentralized AI marketplace and incubator, allows us to have access to world-renowned AI experts from all different fields and really fosters collaboration. We have the Methuselah Fly model. If any of you have ever heard of Michael Rose from UC Irvine, he was my mentor. I studied in his lab. I now take care of the famous Methuselah fruit flies. I've always loved to refer to them as the best high throughput in vivo screening model for aging-related disease. I'm sure most people here know that we share approximately 60% of our genome with fruit flies, and in particular, a lot of the pathways and genes that are involved in aging are conserved across our species. The Drosophila are great because they are a really fast screening model, and it's a good whole systems biology model that we can use to see how a substance affects an organism across their entire lifespan, from everything from breeding to egg laying to development from embryo and all of that. And I have some of the longest lived fruit flies that I think exist. I currently have a stock that is on a 30-week generation cycle. So they live about 7 months, and that is an average lifespan. That's not a maximum lifespan. And so even if we wanted to test substances on them, it would still take a relatively short time relative to other models that are available. Finally, we have the advantage of using crowdsourced human data. We have a partner company called Rejuve AI, and they are a tokenomically incentivized network, and we'll be launching a longevity app later this year. And this is the means by which we are using to crowdsource human data. We currently have about 1,500 people all over the world in beta testing, and this human data will continue to update all of our knowledge graphs that we are using for our integrative AI research. As I mentioned, I'm part of the SingularityNET ecosystem. There's approximately 15 spinoffs currently. Fun fact, this was supposed to be Ben Goertzel up here talking, but you guys got me instead. I hope nobody's disappointed. I left my leopard print hat at home. So really any of these companies could be put in the center of this thing. We are an ecosystem. We all work with the same basic AI tools, and this also contributes to the sophistication of our knowledge metagraphs and fosters collaboration across all different market verticals. And everybody who knows Ben knows that we're on this quest to build artificial general intelligence. That's why there are so many spokes to this wheel. It will eventually be a global brain that can solve all of our problems. So this is the basic Rejuve.Bio AI architecture, the Bioatom space. It's fed by lots of different high-quality data sets. We have genomic data from my Methuselah flies and along with genes and transcriptomes that we have identified that are significant in playing a role in their longevity. We get the Rejuve Network data, public biodata. Currently, we have data from the NANES database. I think the UK biodata was mentioned earlier. And all of this is fed into each other, and the data interacts with each other much the way that a human brain would interact with each other. So drawing inferences, able to generate hypotheses, which, as you can see, would be really beneficial in guiding biomedical research, and in particular, reducing failure rate when it comes to fostering research and predicting what drugs would make it versus which drugs don't across that translational valley of death. You have the Rejuve.Bio application server here. You have encrypted user data that would go in there. Encrypted user data could come from our longevity app with our partnership with Rejuve AI, or from other research institutions using this Bioatom space for their own research applications as well. What I'm going to show you today is our gene annotation service prototype that we have. So this will be a little corner of our Bioatom space. And what you can do is you can input genes that currently you can input genes that you're interested in studying that might be associated with longevity or particular diseases. Eventually, we will have a natural language interface option for this as well, but for now, you need to know specifically what genes you want to look at. So in this case, we have a few genes that we have identified in our fruit flies that have good human orthologs that are involved in aging pathways. You can select what pathways of interest you want to study here. You can select your gene ontology terms, biological processes, cellular components, molecular function. And you can select if you want RNA or non-coding RNA. So in this example, we have a non-coding RNA selected. And so this gives you this massive graph, and you can select different node types and link types between different genes, reactome, metabolites associated with your original input genes that you're interested in studying. This is the complete connection graph for the original input genes. The original input genes are kind of hard to see on here, but they're the little tiny blue dots. But this shows you all the different protein-protein interactions and metabolites that are produced, the various reactive pathways and genes that are associated with your original genes. If you unselect the gene node type, you can curate it just to the genes that you want to look at and kind of remove the associated genes to have a clearer picture of your original genes. And you can explore reactive pathways. If you hover over each node, there will be a link to learn more, and this gets data from our knowledge graph and the available literature that it will allow you to learn more about that particular gene or cellular processes. The reactive pathways are the triangles in there, metabolites are diamonds, and in this example, these are what's associated with the clock gene for circadian rhythm. Zooming in, you can see the particular HSA reactive pathway. And here's a metabolite that's produced by that reactive pathway. Zooming back in and reselecting your gene nodes, zooming back out rather, you can see all the genes that are associated with that particular reactive pathway. In this case, acetylactate histones with gene node types and other genes associated in that reactive pathway. This is a gene ontology graph of the original input genes, so you can go to each gene ontology category. And in this example, all the entities are linked to source database reference in the different Go categories for original input genes, such as biological processes, which are in green. Cellular components are orange, and then molecular function are the yellow bars. So you can see that already this produces a wealth of information right here, and since it's part of our ecosystem, it will be continually updated with new information as people use the Longevity app, as people use this service for their own biomedical research at various institutions. A little bit about what makes the Bioatom space different from other AI out there. We are considered a very advanced neurosymbolic AI framework. It's a dynamic knowledge-based, and it's continually updated, as I've mentioned. It also has a very unique ability to extrapolate pretty reliably from relatively small data sets, which is unique. You don't need huge, massive data sets to get a reliable output. The meta-representational knowledge graph fosters synergistic collaboration with neural nets and other machine learning tools, and integrates varied knowledge graphs under unified architecture, fostering shared knowledge. Our probabilistic reasoning algorithms enable transfer learning from model organisms like flies to humans and across divergent human populations. So really taking all of like the massive amounts of human data that we've collected over human history and animal data and integrating it and actually turning that into usable cross-reference transfer learning to output actionable pathways that can be used for further research. Our neurosymbolic AI integration facilitates symbiotic learning, which is a feature that, like I described earlier, like if a certain AI process gets stuck in our analysis, we have other techniques that can take over. So it's like, if you think of your human brain, if you get in a car accident and you forget, and you don't just forget like half the alphabet if you have a brain injury, it just becomes kind of fuzzy because other parts of your brain kind of take over and fill in those gaps. This AI works in a very similar fashion. As I mentioned before, we have a partnership with our sister company, Rejuve AI, and the CTO of Rejuve AI, Debbie Duong, is here, and she will be on a panel tomorrow talking about the creation of a comprehensive biomarker of aging. Which is really exciting. She's the architect of our app and our generative cooperative networks, and she's brilliant. So make sure you check that talk out tomorrow. This is how we're crowdsourcing our human data to integrate with our animal model data. As I mentioned before, Rejuve AI is tokenomically incentivized and blockchain-based. So this allows people to actually make their data into individual NFTs, and they can decide where their data gets contributed in our research. They also will get compensated via tokens for the launch of any products from the biotech side. So it really is an incredible method of democratizing and decentralizing biomedical research. People will be able to select if they want to participate in various clinical trials through the app. As somebody who's been involved in human research before, I'm just really excited because this is an opportunity for us to have a truly diverse human data set. Most human clinical trials I've run before have skewed wealthy and white. This is a great way to get around that and really have a comprehensive, diverse human data set and allow people to get compensated for the use of their data and keep track of where their data actually goes and have a say over that. We use homeomorphic encryption, which allows that data to remain encrypted so that people still maintain privacy over their data, but we are still able to analyze the data and use it without it being unencrypted. These are my gratitude slides. So this is my team. Dr. Ben Gertzel, of course, Mike Duncan, my CSO, is here. Also notable, Axel Schumacher. He came up with the epigenetic theory of aging. He will be representing us next week at the Dublin Longevity Conference and then at Morton's conference in Copenhagen. He will be there as well. My scientific advisory board, I want to thank all of them for their advice. Obviously, the incomparable Aubrey de Grey is here, who I am delighted to work with. And then that's me. So thanks, everyone. Does anybody have any questions? Yeah. Oh, or you can have this one. You can have this one. You can use that one.
Thanks. Just wanted to follow up on something you mentioned in the beginning about the fruit flies.
Yeah.
Did I hear correctly that you tripled or almost quadrupled the lifespan from the original?
Yes. So Michael Rose started that experiment in 1980, and he took the flies from about living about a month long to living 10 weeks. I took over the flies in 2003, and then I started selectively breeding them again in 2008. So over the course of the last 15 years, I've gotten them to a 30-week generation cycle. Right now, maximum lifespan is about 300 days.
Yeah, so this is amazing. And
yeah.
Did you sequence maybe like intermediate generation to try to see like driver mutations that you can pinpoint to actually, increasing lifespan
We currently did a time point analysis of transcriptome and genome of our super geriatric super Methuselah flies, and we found some interesting things from those. On my dream list of experiments is, of course, to do sequencing at other various time points throughout their lives, in a early life, pre-reproduction, and post-reproduction, and then the geriatric flies. So that's on the docket.
so to be published.
What I would like to
genomic?
I have not published it, no.
Looking forward to that.
Thank you.
Thank you so much.
Thank you. Do we have any more questions from the audience? No? Over here.
Thanks so much. I was wondering also about the Methuselah flies. Mm-hmm. One issue that I often think of with model organisms for aging is that humans may have been optimized by evolution for longevity in ways that some of these model organisms have not. And so we could see diminishing returns with a lot of the things that say double or triple lifespan in C. elegans. So I was wondering, as these flies have been optimized by selective breeding to live longer, do you know if there are diminishing returns for a lot of the things we have used to extend lifespan in Drosophila and C. elegans, like interventions? And would that make them a better model for human aging?
In terms of like diminishing returns, are you referring to like basic life processes such as reproduction and mating and things like that?
I mean in terms of something that, a compound that extends lifespan by, say, 2 or 3 times in C. elegans is unlikely to do so in humans, even if the pathway is conserved and is associated with longevity in both.
Okay, so with my previous company, which is currently being spun into the Rejuve.Bio project, we actually developed a supplement that was supposed to target genes related to Alzheimer's disease and neurological function that we found in our super Methuselah flies. We conducted a Alzheimer's model with fruit flies inducing Alzheimer's and tested our compound. It extended their life and preserved their function in terms of mating and reproduction and egg production. We found a very open-minded neurologist in Newport Beach who allowed us to conduct a human pilot with 50 Alzheimer's patients, adding this supplement to their normal regimen. And the people who fell into the mild and moderate category actually maintained cognition over the course of the 15-month study, and that paper was published in 2018. And so we have, on our shoestring budget that we had at the time, we were able to kind of attempt a proof of concept in terms of showing that particular pathways in the Drosophila that we were studying in this case for neurological function do appear to be conserved in humans. Now, as I mentioned, we were on a shoestring budget, so unfortunately we didn't do biomarker or mechanism of action studies. Everything was functional, cognitive tests that we did with these people who participated in the pilot, but that's about the closest evidence that I have to that makes me still have faith in the Methuselah flies as a model for human aging.
Thank you.
Thank you.
We had a question over here.
My question is on the human clinical studies you're thinking about doing decentralized. I'm wondering if you can speak really quickly to, I obviously see the benefit of those. You detailed some of those in terms of like a broader base of people to draw from, more diverse, ethnically, genetically, sexes between male and female, et cetera. But would you be collecting samples? I'm curious as how you would get quality data from individuals in a decentralized way. It just seems like there would be a lot of challenges related to that.
Yes, so the Rejuve AI company, our sister company, is developing many partnerships with various genomic sequencing companies, metabolite sequencing companies. My dream, eventually I'd like to do microbiota as well. So these people who are already using the app can use their utility tokens that have already been purchased or ones that they earn through contributing their basic data like height, weight, sleep, whatever, can use their tokens for getting these sequencing, these higher order biological tests done. And that data will all be contributed through the app, which will go into our AI platform. Each piece of that data will be an individual NFT that people will be able to monitor how it's used through the blockchain, will be encrypted. And but through the way that we're collecting the data, we can separate out people who might be on certain supplements or people who might have certain factors that might make them a little bit more unique in terms of studying their actual aging or background. So there's lots of like different ways that we can separate people out to actually get like tailored studies. And obviously everybody would have the choice to participate in this study. They don't have to if they don't want to, but it might be worth more utility tokens if they do, which then can be used for other in-app purchases or discounts on certain things and other partnerships that Rejuve AI has made. So it's a full tokenomically incentivized network. So people are, really getting compensated and getting a lot of value for the use of their data. It's not just like blindly giving their data to some study.
Yeah, I think it was more maybe a question on compliance, like ensuring that people are doing the things that you needed them to do as a part of an intervention.
Oh, because it's like it would be self-reported.
Exactly.
Yeah, well, I mean, that's a confounding factor currently. So, I mean, we would hope that people would want to contribute, that the community would be excited enough for the research to happen that they would want to be, honest in that contribution. So, yeah.
Fair enough. Thank you.
Anything else? Okay.
Well, if nobody else has a question, I have one more question. How much longer do your Methuselah bred flies live on average compared to wild type?
About, let's see, wild type are about 40 days currently, and then my super Methuselahs are at a max of like 300.
Wow.
So
related to a question that was asked earlier, have you compared the lifespan increase when you throw in a variety of compounds on these Methuselah flies versus wild type flies to see if you get a similar or less percentage increase? Like if you calorically restrict your Methuselah flies, do they still percentage-wise extend as much as wild type flies?
So the way,
as an example.
No, and I have seen that effect before. In the past, what we've done, we've kind of used the Methuselah flies as like the marker, and so for supplements and compounds that we were testing before with my previous company, we would test them on the wild type flies and see if we could get them to behave like the Methuselah flies. And that's how we screened substances that ultimately led to the Alzheimer's supplement that we tested in a human pilot, and then also tested them in the Methuselah flies and found that it also made them live longer and preserve their function throughout life.
And is there like a Venn diagram overlap of compounds that boost the lifespan of your wild type flies but don't for Methuselah? Or is there a subset that work for both? Or
there were compounds that worked for both. Like one particular natural compound that I'll mention just because it's natural and obviously can't be patented is astragalus root, which is a potent anti-inflammatory and has evidence of being immune system modulating. That was really positive in extending the lifespan of our baseline flies and also had positive impacts on the lifespan of the Methuselah flies at that time. Now, this was like, 10 years ago that I first did those studies. So at that point, I didn't have the super Methuselahs up to like the 30-week lifespan, but it did give them like a good week, 2 weeks of extension in their life.
all right, thank you very much.
Thanks.